Hello everyone, Dr. Devika Rai here. Welcome back to my YouTube channel, Dental Planet. Topic for today is meiosis. Meiosis is also known as reductional division. Why it is called as reductional division? Because if the parent cell has to n number of chromosomes and its daughter cell will have n number of chromosomes, and you can see the reduction of number of chromosomes. So hence it is known as reductional division. Whereas in mitosis, we had seen it is a equational division because the parent cells as well as the daughter cells had the same number of chromosomes. So that's the difference between mitosis and meiosis. So now let's learn meiosis in detail. Meiosis involves two stages, that is meiosis 1 and meiosis 2. Whereas in mitosis, we had only one stage. So first one, that is meiosis 1, under that prophase 1. It is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into leptotin, zygotin, pachytin, diplotin, and diakinesis. So first, leptotin. Initially, chromosomes are seen clustered, whereas in leptotin stage, what happens is gradually the chromosomes will start to disperse and individual chromosomes are seen, and chromosomes are start to get thickening. So that's it about the leptotin. Mainly the chromosomes will disperse and chromosomes will start to thicken. Next is zygotin. Zygotin, what happens is the two chromosomes will start to pair up. So here you can see the two chromosomes. It will start to pair up and the complex is formed. That complex is known as synoptonemal complex. And these two chromosomes are known as homologous chromosomes. So mainly in zygotin stage, we can see the pairing of two chromosomes. That pairing of two chromosomes is known as synapsis. Okay. So that's it about the zygotin. And then pachytin. In pachytin, what happens is, here you can see the two paired chromosome. That is homologous chromosomes. Between homologous chromosomes, you can see the exchange of genetic material. So that exchange of genetic material is known as crossing over. So mainly in pachytin stage, the crossing over takes place with the help of enzyme recombinase. So here you can see the synoptonemal complex between the two chromosomes and the exchange of genetic material at the end of the chromosome, that is crossing over. Okay, so that's it about the pachytin. And then diplotin. In diplotin, what happens is dissolution of synaptonemal complex occurs. Here you had seen the synaptonemal complex. Here you had seen the synaptonemal complex. So that synaptonemal complex is dissolved in diplotin, except at the site of crossing over. Except at the site of crossing over, everywhere the splitting of the chromosome occurs. So that X-shaped structure is known as chiasmata. So in diplotin stage, mainly the dissolution of synaptonemal complex occur except at the site of crossing over, and that structure is known as chiasmata. And in diakinesis, what happens is terminalization of chiasmata. In chiasmata also, what the crossing over site also, it will start to split, and you can also see the spindle fiber formation, and the nuclear membrane will get breakdown, will start to break down. So to sum it up, the prophase 1 stage, starting with the leptotin, mainly thickening of chromosome occurs and individual chromosomes are seen. And then zygotin, the pairing of chromosomes occur, so is known as homologous chromosomes, and synaptonemal complexes formed between the chromosomes. In pachytin stage, the crossing over occurs, exchange of genetic material occurs between the homologous chromosomes. And then in diplotin, dissolution of synaptonemal complex occurs except at the site of crossing over. So that X-shaped structures is known as chiasmata. And in diakinesis, terminalization of crossing over occurs, and mitotic spindles are seen, and nuclear membrane starts breakdown. And next, moving on to the second stage, that is metaphase 1. After prophase 1, metaphase 1. So in metaphase 1, as you had learnt in mitosis stage, in metaphase what happens is the chromosomes will come at the equatorial plate. So here also the chromosomes, homologous chromosomes will come at the center of the cell, that is at the equatorial plate, and the mitotic spindle fibers will attach to the homologous chromosomes. So that's it about the metaphase 1. And then anaphase. In anaphase what happens? The splitting of the homologous chromosome occurs. Okay, splitting occurs. It is pulled towards the opposite poles. So that's the characteristic feature of the anaphase. And then in telophase, separate cell organelles will start to form, separate nucleus will get start to form, and the cytoplasm will start to divide. So that all features is seen in telophase 1. And then cytokinesis occurs. After separate cell formation, cytokinesis occurs. So that's it about the meiosis 1 stage, starting with the prophase 1, and then metaphase, and then anaphase 1, and then telophase 1. And in meiosis 2, what happens is, after separate two cell formation, again these two cells will start to divide. Okay, so in first starting with the prophase 2, in prophase 2 what happens is nuclear membrane will start to disappear, and the spindle fibers will start to form. Chromosomes will get thickened. And in metaphase what happens is, it's just the chromosomes will come at the center, at the equatorial plate, and the spindle fibers get attached to the chromosome at one end and the centriole at the other end. And in anaphase 2 what happens, it's just the splitting of the chromosomes occurs. Okay. And in telophase, again the cell organelles will form, again the nuclear membrane will start to form, and the cell will start to divide again. Okay, so that's it about the telophase. And then cytokinesis occurs. Okay, as I explained earlier, at the end of the meiosis stage we'll have four haploid cells. How, I'll explain. Okay, so if the parent cells has two n number of chromosomes, after meiosis 1 the number of chromosomes will get reduced to n, that is half of the chromosome. And I told earlier that is meiosis 2 is similar to mitosis stage. Okay, so here what happens is there is no reduction in number of chromosomes, and again two cells are formed by each cell. So again the same number of chromosomes will be same, and hence at the end of the meiosis stage we can see the four haploid cells. So that's how cell division occurs, and it is about the meiosis. I hope you all understood this topic. If you all feel it's useful and informative, then hit a like button and subscribe my channel. Share the video as much as possible. Thank you.